Hello, welcome to this Elevate Education presentation. Today we're going to be looking at how you can calculate the magnification of an image. The keywords we're going to be using are micrometer and nanometer. There are three parts to today's video. First of all, we're going to look at the units of measurement commonly used in biology when looking at cells. Secondly, we're going to look at how we can calculate magnification from looking at a biological drawing. And thirdly, we're going to have a look at some exam style questions. We're all comfortable with the idea that we use centimetres and millimetres to measure distance. However, when we're considering things that are very small, such as cells, or even smaller, such as cell organelles, these units of measurement, this scale, becomes very hard for us to work with. To work with 0.00001 of a millimetre is a very difficult number. So, when we're looking at very small things, there are some other units of measurement that we use. The ones that we need to be aware of for cell biology are firstly micrometers and secondly nanometers. A micrometer is 1,000th of a millimeter and a nanometer is in turn 1,000th of a micrometer. If we wish to convert centimeters to millimeters, we should know we times them by 10, but to convert millimeters to micrometers we have to multiply the millimeters or measurement in millimeters by 1,000 and the measurement in micrometers by 1,000 to turn it into nanometers. To turn nanometers back to micrometers, and in turn micrometers into millimeters, we do the reverse operation, we divide. So, if we look at 1 centimetre, we can see that 1 centimetre is in fact 10 millimeters, which is the same as 10,000 micrometers, or 10 million nanometers. The unit of measurement that we are going to be most interested in for looking at cells and cell organelles is that of micrometers. So we need to be confident that we know how micrometers relate in size to both centimeters, millimeters and nanometers, and how we can transfer between those units. In the rest of this video we're going to be looking at how to calculate magnification. To do this, there are three terms that we need to understand. The first one is that of image size. When we're talking about image size, we mean how large an image is. So, if we are looking at an image, a picture of a cell, the image size is how large the picture is. In contrast, actual size is a measure of how large or how big something is in the real world. So if we're thinking about a cell, the actual size of the cell is how big the cell really is. Magnification is a term that refers to how much larger an image size is than the actual size. How many times the image has been enlarged. If we're going to understand how to calculate magnification, we first need to understand how these three terms relate to each other. I've shown them on the screen in this triangle, which is a formula triangle, a common way of showing formulas. The horizontal bar represents dividing, and the x between actual size and magnification represents multiplication. The important thing to notice about the image sizes and the actual sizes that we're working with today is that they're both measured in micrometers. When we're going to be calculating magnification, we're always going to work with the image size and the actual size in micrometers. Therefore, if we have a measurement that's in a different amount, if we're given an actual size or an image size in millimeters or nanometers or centimeters, we're going to need to convert it to micrometers before we can proceed with any calculations. The way we use this triangle, and we cover up with our finger the thing that we're trying to work out. So, if we're trying to calculate the image size, how large something is going to appear in an image, we can cover up image size in our formula triangle, and that will tell us what we need to work out. So we can see image size is equal to actual size multiplied by magnification. If we're trying to calculate magnification, in turn we cover up magnification, and we can see magnification is equal to the image size divided by the actual size. And if we're trying to work out how large or how big something is in the real world, its actual size, we need to divide the image size by the magnification. Here is a typical exam question. We're being asked to calculate the magnification of the animal cell in this image. We know it's an animal cell because it's only got one membrane around the outside. If we look underneath the cell, just below it, we notice there's a black bar. This is called the scale bar. Underneath the scale bar is a number, in this case 5 micrometers. This tells us that if an object is the length of this black bar in the diagram, it has a length in the real world, an actual length of 5 micrometers. The first thing we need to do is work out which formula we need to use to calculate magnification. So, we look at our formula triangle, we cover up magnification with our finger, and we see that magnification is calculated by dividing the image size in micrometers by the actual size in micrometers. If we look at the diagram, we know the actual size of something already. We know the actual size of the scale bar. We know that that black line represents an actual size of 5 micrometers. So we can work out the magnification of the image by working out how magnified, how many times enlarged the scale bar is. So the magnification is equal to the image size divided by the actual size. The actual size we've just said is 5 micrometers. Next we need to work out how large the scale bar is in the image. And the way we do that is we get our ruler and we measure it. But our rulers don't measure in micrometers, which is the unit we have to use, they measure in millimeters. So, our scale bar is in fact 17 millimeters long. Before we can work out the magnification, we have to convert that into micrometers. So to convert millimeters to micrometers, we have to multiply by 1,000. So we end up with 17,000 divided by 5, which tells us the magnification of the cell in this diagram is 3,400 times. The next way the example can ask us to work out magnification is they could give us a question such as this. We're told that organelle M is 2.5 micrometers long. We have to use that information to calculate the magnification of the image. We've been asked to work out the magnification. So the first thing we do is we write down our formula. Remember, we've checked our triangle. It's image size in micrometers divided by the actual size in micrometers. We're told by the question that the length of that mitochondria, the actual size of it, is 2.5 micrometers long. But to work out the magnification, we also need to know the actual size. So we need to measure the same measurement in the image. So we need to get our ruler and measure how long our mitochondria is in our image size in millimeters. In this case, it's 8.5 millimeters long. Again, we can't use millimeters in our calculation where everything has to be in micrometers. So we turn our millimeters into micrometers by timesing them, multiplying them by 1,000. So we end up with 8.5 times 1,000 divided by the actual size of 2.5 micrometers, which gives us 8,500 divided by 2.5. So we end up with a magnification of 3,400 times. Those are the two ways that the example can ask us to work out magnifications. Look at this question on the screen now. On the right hand side is a picture of a plant cell. We're asked to calculate the magnification of the image. There's a scale bar at the bottom and a ruler showing a distance of 12 millimeters. Pause the video, see if you can use this information to work out the magnification of the image. Hopefully you worked out that this image has been magnified by 1,200 times. It's 1,200 times larger than it is in real life. And you went through the steps that we saw. You divided image size by actual size. You multiplied the millimeter measurements by 1,000 to turn them into micrometers. Today we've looked at how to work out magnification in biological drawings. In our next video we're going to look at how to work out actual size and image size from similar drawings. If you want to have more practice working out magnification from biological drawings, please visit our SlideShare site for some questions. We can be found at www.slideshare.net/elevateeducation, all one word. Alternatively, if you'd like to find out about other areas of the AS biology course, please keep watching our channel and more videos will be posted soon.